student. Today let us discuss about the topic that is theories of developmental biology. Mm, in the previous class, we have to discuss that is the introduction to developmental biology. In this introduction to developmental biology, we have to study that is the definition of developmental biology, difference between developmental biology and embryology, and stages of developmental biology, historical review of developmental biology, and that is the scope of developmental biology. Now today we have to start that is the theories of developmental biology. In the developmental biology there are 10 theories are there, that is theory of preformation, theory epigenetic theory of pangenesis theory, recapitulation theory, gene plasma theory, mosaic theory, regulated theory, gradient theory, and theory of organizers. And another one theory will come, so that is the von Bayer's law theory. Actually there are 10 theories are there. In these 10 theories we have to classify it into two types, that is on the basis of historical and on the basis of experimental. These theories have to be grouped, that is 5 theories are comes under the historical basis and 5 theories are comes under the experimental basis. That is theory of preformation, theory of epigenetic, theory of pangenesis, recapitulation theory, and von Bayer's law theory. These theories are comes under the historical basis, and the remaining the 5 theories, that is gene plasma theory, mosaic theory, regulated theory, gradient theory, and theory of organizers. These theories are comes under the experimental basis. Means those theories they have to conduct on experiment and they have to prove, and those theory only on the basis of history they have to prove, that is the historical basis theory. Now let us discuss about one by one these theories. First we have to start with the theory of preformation. What is the theory of preformation? This theory of preformation was proved by, that is Marcello Malpighi, to explain the development. According to him, what said this theory means, pre means first, that is before the formation what will happen, that is it is an development of an organism. According to this theory, the gametes contain in them a mini preformed embryo. In the gamete, that is mini preformed embryo is present. Means if the womb is contain a miniature adult, that we call as homunculus. If the sperm is contain miniature adult, that we call as animalculus. In the case of man, we have to taken the miniature organism was named as homunculus. Means in the home, if in the womb, if it is contain the preformed embryo, that we call as homunculus. If the inside the sperm, if the preformed embryo is present, that we call as animalculi. But in this case, the egg develops into an animal without the assistance of spermatozoon. If the egg can develop without fertilization, means the embryo cannot be preformed inside the spermatozoon. Therefore, actually this theory was not at all accepted. Here there is already I have told that. And the another one scientist is also believed that is Leeuwenhoek and R. Zucker also they have to share that the whatever the mini preformed embryo is present in the sperm, that they were called as animalculi, and these scientists were called as animalculist scientists or spermicist. Here we have to see in the diagram, actually this is an sperm. In the sperm head, that is preformed embryo is present, that is means homunculus is present. This is about the preformation theory, but actually this preformation theory was not at all accepted. Next come to the theory of epigenesis. Theory of epigenesis, it was proposed by, that is Wolf, Friedrich Wolf, and it was actually based on the observation of the development of the chick embryo. This theory states that there is no preformed embryo inside the egg, where there is no miniature adult that are present inside the egg. The egg, what the egg is formed, that is only formed by the granular substances, and these granular during the development, actually these granules at the time of development they are arranged from the cellular layers, those cellular layers where they were called as germinal layers, and those germinal layers, that is, they grow thick in some parts and they grow thin in some parts and make them as a pocket like structure and become an body of an embryo. This is about the theory of epigenesis. Now come to the, in the year 1866, the another one theory was proposed, that is a pangenesis theory. Pangenesis theory, it is the developmental theory of heredity. In the Darwin, the Charles Darwin, he suggested that all the cells in an organism are capable of sending minute particles that called as gemmules, which are able to circulate throughout the body and finally congregate in the gonads. Means what it means that the Darwin that assumed that each cells of the body of an organism that they have to produce a minute copy of its own cells that called as gemmules, and these gemmules are that they have to pass into the bloodstream, and those they are carried by the blood and deposited in the testis and ovary. Thus the gonads are we call as the storehouse of the various types of gemmules, and these gemmules are then given to the gametes. The young one formed from the gametes should be having all the gemmules of the parents. Actually, this theory supports the idea of inheritance of acquired characters, that is Lamarckism is the same as that of this theory was. No, this theory proposed by that is Darwin. Actually, it was mainly based on the cell theory. That is, cell theory was proposed by that is Skeldon and Scaven theory. This is about the theory of pangenesis. Next come to the theory of recapitulation. It is we also called as biogenetic law theory. Actually, this theory was proposed by Ernest Haeckel and as well as Muller. It is actually modified theory of the von Bayer's law theory. It is actually based on the contribution of evolutionary theory. This theory states that higher animals in their development passes through their stages which are similar to the other stages of lower animals which were their ancestors. Means here again the ontogeny is recapitulates the phylogeny. In other words, we say that is whatever the ancestral characters they are reappear in the developmental stages of an individual. Thus the biogenic law or recapitulation theory actually state that is embryonic stages of the higher animals, those resembles same as that of the adult stages of its ancestor, that called as recapitulation theory or biogenetic law. For example, you have to say for example you have to take that is the frog. In the ontogeny of frog, there appears a tadpole larva. This larva, in every aspect, we have to see that it resembles like as that of an adult fish. According to the biogenic law, the fish it is an ancestor of the amphibians. This is why that the development of amphibian there is a fish-like tadpole stage. Therefore, this is same as that of the whatever the amphibian larva is look like same as that of the fish like in the tadpole stage. This is about the biogenic law or recapitulation theory. Here we have to see that is in the diagram, that is fish is there, salamander is there, turtle and chicken and rabbit and human. These are figures. They have to show that the recapitulation theory or biogenetic law theory. Next, we have to discuss about the von Bayer's law theory. Von Bayer's law, it is actually he was the founder of comparative embryology. Here there are four concept they have given. Actually, these are the four principles are there. What they have to say the principle means, the first principle is that is the general features of a large group of animals appear earlier in development than the specialized features in a small groups. Means it means the more general features of any large group of animals they appear in the embryo earlier than the more special features. This is the first one law principle. Or second one is after the more general features those that are less general arise until the most special features appear. This is the second principle. Third principle is here the embryo does not pass through the adult stages observed in lower animal. That is here ontogeny does not recapitulates the phylogeny. Means the embryo of different species are more alike than the adults and progressively from each other during the ontogeny. And the fourth come to the early embryo shares characteristics in common and become more and more divergent as development proceeds. It means whatever the embryo of a higher species may resembles the embryo of a lower species, but the embryo of the higher species never same as that of the adult of the lower species. Example, you have to take the embryo of a frog resembles the embryo of the fish. This is the fourth one law. For example, you have to take in overall of the four principles, that is in the development of chordate, you have to say the more general features like brain, notochord, some muscles they have to develop first. But after the features we have to taken as the limbs are developed, feathers in birds are developed, hairs in mammals are developed. They are depends on the finally of the specific features of their characters distinguish their families, genera and the species will be developed. This is about the own Beer's law. Next come to the germ plasma theory. This theory was proposed by the August Weismann. He was explain theory on heredity and the development of an animal. According to this, the body of an organism, it is formed of two types of cell, namely that is the somatic cells and another one is the germ cells. Somatic cells, those cells they are the main body bulk of the somatic cells, and those somatic cells they have to disappear with the death of the animal. Therefore, there is no any change affecting the cells. It is not heritable. But those gamete cells are there, germ cells are there, they are carried to the generation after generation. So the change affecting these cells are they are inherited. For this germ plasma theory, according to Weismann, each blastomers that will be contain only one type of determinant. If a blastophore is separated from an early cleavaging embryo, then the remaining cells should develop contain only a particular type of organ and not a complete embryo. But now it has been proved that blastomer separated from, that is 4 cell stage or even 8 cell stage, 16 cell stage or 32 cell stage of the embryo develops into a complete larva. This is about the germ plasma theory. Next come to the gradient theory. What do you mean by gradient theory? Actually, original gradient theory was given by, that is the T. Bowery and M.C. Child, and actually this theory was given by T. Bowery, but it was supported by M.C. Child, and after this theory we call as metabolic axial gradient theory, because Bowery he was demonstrated that he has conducted the experiment on sea urchin, and he has influenced them. He has said that in the egg is having the animal pole and vegetal pole, and animal pole is dominant and it influence gradually and decrease towards the animal pole. But this was the fundamental idea for the gradient theory. But however, Boyer was not able to give the proper reason about this gradient theory. But later they come to the M.C. Child, and he was strongly supported this gradient theory with his experimental evidence, and he said that what he said that means whatever the animal pole is there, that is different from the vegetal pole in its rate of metabolism. When the rate of metabolism is higher at the animal pole and it is gradually decreases towards the vegetal pole, where there is the rate of metabolism is lesser. Therefore, this theory we call as metabolic axial gradient theory. And there are this gradient theory was highly modified by, there are many embryologists, that is hostids, runners, they are called as double gradient theory. They explain the egg development on the basis of animal pole gradient and vegetal pole gradient, because in the animal pole gradient there is a tryptophan, that is amino acid is present, and the vegetal pole gradient, that is in the presence of nucleotide is present. Therefore, the animal gradient is highest at the animal pole and fades towards the vegetal pole, whereas vegetal gradient is the highest at the vegetal pole and fades towards the animal pole. This is the presence of the, that is the tryptophan and nucleotide. This is about the gradient theory. Next we come to the Mozart theory. No, Mozart theory actually developed by, that is the proposed by, that is Raux. The Mozart theory actually it states that every part of the egg is predetermined to develop into a particular organ. Every part of the egg is predetermined. The Tampering of any part of such a predetermined would result in a corresponding defect in the embryo. This type of development we also known as determinate development. Here we have to take the experiment. How they have to conduct, that is, you have to take the fertilized egg. After they undergo cleavage, become two cell stage. In this stage, William Raux, he destroyed one of the two blastomers of the frog with a red hot needle. The embryo that was derived from the surviving blastomers was defective in many respects, that is, surviving blastomers is there, that will be developed into a half embryo. Means here what is that, whatever is the dead hot needle that will be that the dead tissue, it whatever the hot needle is injected, that will become the dead tissue, and another one becomes the living tissue. Means the half will be destroyed and another one, that is the half, will be developed into the normal stage, that is the half portion will be developed into the embryo. And next come to the regulative theory. What is regulative theory? Actually, regulative theory is different parts of the eggs are not predetermined. They have the potentiality to develop into any part of the future embryo. If a portion of the egg is destroyed or removed, the remaining portion of the egg reorganizes to develop into a complete normal embryo. Therefore, this type of development is known as indeterminate development. Here the Drich, according to Drich, he is taken as if a small piece of an egg is removed or destroyed, the remaining portion of it reorganized to develop into a complete normal embryo. Such a type of development is said to be regulative development or regulative theories. Now come to the Sperman theory. Sperman theory is most important theory that we have to observe in the Sperman Mangold experiment, because in this Sperman, he has conducted experiment on the formation of eye and neural tube. On the basis of these results, he postulated that during development, due to these interaction between different parts of the embryo, the parts develop in their proper position and regulative their relative size. Here, Sperman has conducted one experiment on the frog, that is, he demonstrated that the dorsal lip of an early gastrula, they have to grafted into another one embryo. The graft along with the surrounding tissues develops into a whole secondary embryo. On the other hand, if the dorsal lip of an early gastrula that he has to destroyed, but the embryo fails to develop. Therefore, thus it clear that the dorsal leaf organizes the entire process of development. Sperman named this tissue as an organizer. For this experiment, he was awarded the Nobel Prize for his discovery, and that tissue we call them as an organizer. Organizer is defined as region or group of cells in an embryo that can both induce and pattern and adjusted embryonic cells. It is also known as inducer, and its morphogenic effect is known as induction. In the next class, we have to study it in detail, that is embryonic induction and organizer phenomenon topic. This is about the Sperman theory. These all are theories we have to discuss in today, that is, there are total 10 theories are there, and each theories they have given the different idea and different concept. Thank you.